Imagine for a second, you're trying to figure out a highly coordinated, completely encrypted communication network that's spread across this massive city.
Right, like a sprawling metropolis.
Exactly. And, you know exactly who lives in the city. You have the demographics down perfectly. But your only way of analyzing their actual network is to take like a satellite photo, draw these arbitrary circles around different neighborhoods, and then just guess who is talking to whom, based entirely on how close they are standing to each other.
Which is a pretty terrible way to intercept a message.
It's awful. I mean, you see a sender, you see a potential receiver nearby, and you just assume, oh, a highly classified message was successfully transmitted and they acted on it.
Huh.
But that assumption, that proximity just automatically equals a completed transaction that has been the Achilles heel of spatial biology for years. So, welcome to the deep dive. Today, we're tearing into the stack of sources you sent over, specifically focusing on this freshly published Nature Communications paper. It introduces a computational framework called Renoir.
And it is a crucial paper because it addresses exactly that blind spot you just described. Our mission today for this deep dive isn't just to look at, another tool that catalogs cell types. We're looking at a fundamental shift in how we decode functional geography in our tissues.
Yeah, moving beyond just the basic roster of cells.
Right. I mean, single-cell RNA sequencing already does the roster brilliantly, but we need to map exact spatial communication domains where we can mathematically prove a signal was actually sent, received, and this is the key part, actively altered the receiving cell's behavior. This is fundamentally changing our approach to mapping neurodevelopment and, maybe more critically, how we dismantle the architecture of tumors.
Okay, let's unpack this eavesdropping problem first, because when you look at the progression of spatial transcriptomics over the last few years, we've obviously moved past that old blender approach, right?
At a blender approach, yeah.
Right, like with bulk or even single-cell RNA-seq, where you just take a beautifully organized tissue, toss it in a blender, and sequence the smoothie. Preserving the spatial coordinates using platforms like 10x Visium or Xeni, I mean, that was a massive leap.
It was huge.
But reading through the background in your sources, it's honestly glaring how simplistic the early analytical tools were.
Yeah.
I mean, they relied almost entirely on ligand-receptor co-localization.
Exactly. The assumption baked into earlier tools, like cell phone DB or earlier iterations of spatial analysis, it was strictly proximity-based.
Just guessing based on neighbors.
Right. They look at a spatial transcriptomics slide, they identify the mRNA transcript for a ligand the message in one spot, and then they identify the transcript for a corresponding receptor the mailbox in an adjacent spot. And they just score that as an active cell-cell communication event.
Which is just a structurally flawed way to look at biological signaling. Let's upgrade the classic metaphor here for everyone listening. It's not just dropping a flyer in a mailbox. I mean, it's like assuming that just because I dropped a flyer in your mailbox, you automatically read it, changed your entire worldview, and bought my product. We don't actually know if the message did anything.
That's a perfect analogy, or think of it like a cryptographic key sliding into a lock. Just because the key fits in the lock doesn't mean the engine started.
Right, the car could be out of gas.
Exactly. The ignition wires could be cut, or the computer could be overriding the start sequence. You don't know the car is actually running unless you hear the engine revving, which in biology is the downstream gene expression.
Okay, that is the precise mechanistic gap.
It is. And biology is just rife with examples where the key fits. But the engine does something completely unexpected based on the context. The paper actually highlights tumor necrosis factors, or TNFs, to illustrate this perfectly.
Oh, the TNF example. Walk us through that.
Sure. So a TNF ligand can bind to TNFR1 or TNFR2. Both are totally valid receptors. But depending on the specific internal machinery of the receiving cell, that single ligand can trigger wildly divergent downstream cascades.
Like totally different outcomes.
Night and day. It might trigger the NF-kappa B pathway for survival and inflammation, or
it could just tell a cell to die.
Exactly. It might trigger apoptosis, cell death. So if your algorithm only scores the ligand and receptor being near each other, you are completely blind to the actual biological outcome.
You're essentially logging a massive false positive for a pathway that might not even be active. And, if you are trying to design a targeted drug to interrupt a specific cancer pathway, relying on a map full of false positives is just disastrous.
It's a waste of billions of dollars in drug development.
Exactly. You don't just want to know if the lock was turned. You need to know exactly which genes were transcribed as a result.
Right.
So let's do a quick 5-minute recap, a little bullet summary of where we are so far for you listening. Bullet 1, older tools just looked at proximity. Sender near receiver equals communication. Bullet 2. That's a bad assumption because having the key in the lock doesn't mean the engine is running. And bullet 3. To cure diseases, we need to know what the cells are actually doing, not just who they're standing next to.
That sums it up perfectly.
Awesome. So that leads us seamlessly to how this new tool, Renoir, actually solves this. It shifts the focus entirely from ligand-receptor pairs to charting what they call ligand-target activities.
Right. Renoir traces the signal all the way to the transcription of the target gene in the receiving cell. To do this, it calculates what the authors call a neighborhood activity score across the entire spatial topography of the tissue.
Okay, but the mathematical architecture underneath that score is what really sets it apart. Because as we know with technologies like Visium, a single spatial spot on a slide, which is usually about 55 microns wide, it's rarely single-cell resolution.
Oh, definitely not. You might have 5 to 10 completely distinct cells mashed together in that tiny spot.
Right, which introduces a massive deconvolution problem.
Yeah.
Let me play devil's advocate here. If you have a mixture of, say, fibroblasts, macrophages, and epithelial cells all crowded in one single capture spot, how on earth does Renoir isolate the specific signal from the noise? It can't just look at the total mRNA soup for that spot, right? It has to mathematically unmix the residents of that specific neighborhood first.
It does. It tackles that deconvolution problem really aggressively before it even attempts to score the communication.
Ow.
So Renoir integrates the spatial transcriptomic data with annotated single-cell reference data sets. It essentially estimates the precise abundance of every specific cell type within that single 55-micron spot. But it doesn't stop there.
Here's where it gets really interesting.
It cross-references the single-cell data to explicitly verify receptor expression constraints.
Yes. This is a critical step to highlight. Renoir basically runs a Boolean check. A true or false. It asks, does the proposed receiving cell type in this specific spot actually physically express the functional receptor required for this ligand?
Right, and if the single-cell reference data says no, Renoir just zeroes out the interaction. It's done. It doesn't care how much ligand is floating around in the neighborhood. If the cell doesn't have the hardware to receive it, the algorithm kills the score.
It's like sending an email to someone who doesn't even own a computer. The message is there, but there's no inbox.
Exactly. That receptor constraint dramatically prunes the mathematical search space. It eliminates a vast swath of biologically impossible false positives. But the real engine of Renoir, the way it calculates that neighborhood activity score, relies heavily on information theory.
Okay, yes.
Specifically, it utilizes Shannon entropy and mutual information to quantify the relationship between the ligand and the target gene expression.
Let's unpack how Shannon entropy is deployed here, because it's a brilliant application of information theory to biology. For you listening, think of entropy, in this context, as basically a measure of uncertainty, or chaos.
Chaos is a good way to put it.
Right. So if you look at the expression level of a target gene across a whole tissue slide, it might look totally random, chaotic, highly variable, that is high entropy. But Renoir calculates the mutual information between the spatial distribution of the ligand and that target gene.
Exactly. It asks a really fundamental statistical question. If I know the concentration of the ligand in a specific spatial neighborhood, how much does that reduce my uncertainty about the expression level of the target gene in that same neighborhood?
Oh, wow. So if the presence of the ligand tightly predicts the activation of the target gene, the mutual information is high, the uncertainty drops. The chaos goes away because there's a clear pattern.
You've got it. And Renoir calculates this while mathematically accounting for spatial lag.
Wait, what does spatial lag mean here?
It means it isn't just looking at the isolated spot itself. It's weighing the diffusion of the ligand into neighboring spots, creating this localized spatial network. It combines all of that, the mutual information, the cell type deconvolution, and that strict receptor constraint logic to generate a highly specific, spatially resolved score for every single interaction.
And then it uses those scores to cluster the tissue into distinct spatial communication domains.
Exactly. It maps the functional geography.
So it's no longer just telling you, hey, there's a tumor over here and healthy tissue over there. It's identifying distinct micro-neighborhoods defined by the specific dialect of chemical signals being exchanged.
Yes.
Now, all this math, the deconvolution, the mutual information, the Boolean checks, it sounds bulletproof in theory. But when you were reading through the benchmarking section of the paper, The way they actually validated this against existing tools was fascinating.
It really was.
Because they didn't just throw it at messy clinical data right away, right?
Right.
I mean, they built ground truth simulations.
Right, because rigorous benchmarking is the only way to validate a tool this complex. They evaluated Renoir against older tools like Komatmuth, Slurn, and Spatial DM. And they used simulated spatial transcriptomic datasets.
What kind of tissues did they simulate?
Specifically, they modeled human intestine, breast cancer, and the prefrontal cortex of the brain. And because it's a simulation, the researchers knew the absolute ground truth. They knew exactly which cells were mathematically programmed to communicate with each other.
Which allows you to objectively measure precision and recall. And the results exposed the exact flaw in proximity-based tools that we were just talking about. There's one interaction in the breast cancer simulation that perfectly isolates why Renoir's receptor constraint logic is totally non-negotiable. It involved the IFNG ligand and the J-chain target gene.
Oh, the IFNG J-chain dynamic, that is the perfect test case.
Walk us through what happened there.
So in their simulation, natural killer T cells and CD8 plus T cells were the designated senders. They were actively secreting the IFNG ligand.
The message.
Right. And the designated target for the simulation was the J-chain gene, which is robustly expressed in B cells and dendritic cells. Now, biological context is key here. B cells inherently lack the receptor to process the IFNG signal.
It's a biological dead end. The B cell literally cannot transduce the IFNG signal to activate J-chain. It's that email to a guy without a computer.
Precisely. But when you look at the output from Komatmuth, Slurn, and Spatial DM, they all hallucinated a massive interaction.
Just hallucinated.
Completely. They saw the high concentration of IFNG, they saw the B cells expressing J-chain in the same spatial vicinity, and their algorithms just lit up the map with false positives. They completely ignored the biological reality that the B cells lacked the receptor to bridge that gap.
Wow. But Renoir caught it.
Renoir integrated the single-cell reference data, saw the missing receptor in the B cell profile, and mathematically vetoed the interaction. It returned a 0 activity score for those areas, perfectly matching the ground truth.
It's a stark demonstration of why these algorithms need strict biological guardrails. And the researchers didn't baby the algorithm either, did they? They subjected it to some severe degradation testing.
Oh, they tortured it. They downsampled the RNA sequencing depth by up to 40%. They injected significant noise into the cell type annotations, intentionally mislabeling proportions to see if that deconvolution engine would just break. And Renoir maintained high structural similarity to the ground truth, even with severely degraded inputs.
Okay, let's do another quick summary checkpoint for you listening. We've got our problem, and now we have our solution. Bullet 1. Renoir uses math, specifically mutual information, tracking the drop in uncertainty to see if a message actually caused an action. Bullet 2. It runs a strict true-false check to ensure the receiving cell actually has the right receptor. And Bullet 3. When tested against older tools and simulations, Renoir caught biological impossibilities that the others missed, even when the data was degraded by 40%.
Perfect recap. And that robustness, passing those stress tests, that is what gives you the confidence to deploy it on actual noisy clinical samples.
Which brings us to the first real-world application in the paper, mapping the adult mouse brain using 10x Visium data. Eager to dive into this one.
The brain is basically the ultimate test for spatial complexity. When they ran Renoir on the coronal sections of the mouse brain, it autonomously identified 20 distinct spatial communication domains.
Wow. 20?
Yeah. And what is striking is that the algorithm has absolutely no inherent knowledge of neuroanatomy. It doesn't know what a brain is.
Right. It's just looking at numbers.
Exactly. Yet, purely by clustering the localized cellular conversations, the domains it generated perfectly aligned with the known anatomical structures, the cortex, the hippocampus, the thalamus.
It essentially reverse-engineered the anatomical map strictly by eavesdropping on the chemical signaling pathways. Like, it localized MAPK signaling predominantly in the cortical layers and pinpointed neurotrophin signaling tightly within the hippocampus, which perfectly tracks with the region's role in plasticity and memory formation.
Exactly.
But the granularity of the astrocyte data, that is what really stood out to me.
Yes. The astrocyte heterogeneity they uncovered is a prime example of why spatial context is literally everything. We often treat astrocytes as this monolithic cell population, just support cells. But Renoir showed that their communication networks are fiercely localized.
Like having different regional access.
Precisely. Telencephalic astrocytes located in the cortex were actively utilizing the VEGF ligand to signal excitatory neurons, specifically in layer 6B.
But if you move just a few millimeters over to the thalamus...
Right, the diencephalic astrocytes are running a completely different operating system. They were using the AC ligand to communicate with inhibitory neurons. Same broad cell class, entirely different functional dialects dictated strictly by their spatial zip code.
That extreme localization of signaling is precisely why the blender approach of bulk sequencing fails us. You just see a mix of VEGFA and ACT and have no idea they were entirely separate regional conversations.
You'd be totally lost.
And while that level of detail is fascinating in a healthy brain, the clinical implications become massive when we apply this framework to oncology. So let's talk about how the researchers transitioned from the mouse brain to mapping human triple-negative breast cancer, TNBC.
TNBC is aggressively heterogeneous. It is notorious for its complex tumor microenvironment. Using Renoir, they stratified the TNBC tissue into 6 distinct communication subdomains.
And again, these computationally derived domains mirrored the pathologist's visual annotations, right? Like differentiating the dense tumor core from the invasive edge and the surrounding stroma.
They did. But Renoir allows us to see the architectural strategy of those regions. Let's look at what was happening at the invasive edge. They designated it as Domain 1.
Okay, Domain 1.
This area was heavily populated by invasive cancer cells, cancer-associated fibroblasts, and macrophages. Renoir isolated an intense, highly localized upregulation of CCL2 signaling in this specific niche.
And for context, CCL2 is a really potent chemokine. Its primary function there is to recruit and reprogram tumor-associated macrophages to support metastasis and build new blood vessels, angiogenesis.
Exactly. So we are looking at the exact spatial coordinates where the tumor is actively recruiting its supply lines. It's the logistical hub of the tumor.
It's building an army.
Right. And in contrast, when Renoir analyzed other specific subdomains deeper within the tumor architecture, it mapped highly active WNT signaling and intense IL-6-JECTA3 pathway activation.
Both of which are deeply immunosuppressive pathways.
Yeah.
The IL-6-JECTA3 axis in particular, that is a classic mechanism tumors use to induce exhaustion in T cells and basically tell the immune system to go to sleep.
Exactly. Renoir didn't just find these pathways. It showed us the blueprints of the chemical walls the tumor is building. It visually maps the specific micro-neighborhoods where the tumor has successfully established an immunosuppressive shield.
So you can actually see the spatial boundaries where the immune system is actively being shut down.
You can see it clear as day.
Which radically changes how you approach therapeutic design.
Yeah.
You realize that delivering a systemic immunotherapy, just pumping it into the bloodstream, it might fail simply because it cannot penetrate the specific communication domains where that IL-6 shielding is thickest.
Exactly. We are moving from identifying general targets to dismantling the tumor's networks block by block.
Block by block. I love that. And that concept of tumors building specific microneighborhoods perfectly bridges into the final and, frankly, most compelling validation detailed in your sources. The investigation into the onco-fetal niche in hepatocellular carcinoma or liver cancer.
This is where they pushed Renoir to its absolute limits. They utilized Visium HD and true single-cell spatial technologies like COSM-X and Xenium.
So, super high resolution.
The highest. They started by analyzing healthy human fetal liver development. During fetal growth, the rapid expansion and organization of tissue require massive, highly orchestrated communication.
Right. The cells have to tell each other exactly how to build the liver from scratch.
Exactly. And Renoir successfully mapped those developmental networks. It identified a critical interaction between Kupffer cells, which are specialized macrophages in the liver, and hepatocytes, specifically driven by the PLG ligand binding to the Marco receptor.
Okay, PLG binding to Marco.
Right. That PLG-Marco axis is foundational for healthy hepatic regeneration and tissue modeling in the fetus.
Got it. So they have the healthy map.
Having mapped that healthy developmental baseline, they then deployed Renoir on hepatocellular carcinoma samples, liver cancer. They were specifically hunting for the onco-fetal ecosystem.
Okay, let's explain the onco-fetal ecosystem for a second because it sounds like sci-fi. This is a phenomenon where tumor cells effectively hijack silenced developmental pathways, right? They basically revert to a fetal-like state to drive aggressive, unchecked growth and evade the immune system.
That's exactly it.
It's like the tumor is accessing a locked, archived zip file of genetic programs from before you were even born, just to survive.
It's terrifying, but it's brilliant biology. And Renoir successfully identified the known onco-fetal interactions, tracking the resurgence of VEGF-A and CXCL12 pathways within these specific tumor niches.
But a computational tool's true value isn't just in confirming what we already know, right? It's in generating novel, testable hypotheses. Did Renoir find something new?
It did. Renoir isolated an interaction that had not been mapped in this context before. Right in the heart of the onco-fetal niche, Renoir detected a dense communication hub centered around the secretion of IL-6 by the tumor cells.
IL-6 again.
Yes. But the critical discovery was the specific target of that IL-6 signal. Renoir predicted it was actively binding to a population of MUC6 plus bipotent hepatocytes.
MEC6 plus bipotent hepatocytes. These are stem-like, highly plastic liver cells, right?
Yes, they are very adaptable. And Renoir's mutual information algorithms predicted that the IL-6 ligand was not just binding to them, but actively triggering the expression of PTGS2 in those bipotent cells.
Okay, let me pause here. PTGS2 is a critical enzyme in the prostaglandin biosynthesis pathway. It is heavily implicated in driving tumor progression and inflammatory signaling.
Exactly.
So the algorithm is predicting that the tumor is actively secreting IL-6 to recruit and essentially corrupt these stem-like cells, forcing them to activate a pathway that accelerates the cancer.
That is the prediction, a highly specific mechanistic prediction based purely on spatial math.
And what elevates this paper from just a fascinating computational exercise to a landmark study is that the researchers didn't just publish the prediction and walk away.
No, they didn't. They validated it in vitro.
The mic drop moment.
It really is. They moved from the in silico model, the computer, straight to the wet lab. They cultured HepG2 human liver cancer cells to serve as the model for this onco-fetal environment.
Right.
And based entirely on Renoir's spatial mapping, they introduced recombinant IL-6 protein to the culture at increasing concentrations.
They basically recreated the exact signal the algorithm overheard in the tumor microenvironment. They dumped the IL-6 text message into the petri dish.
Exactly. And the biological response was definitive. The introduction of IL-6 induced a strict dose-dependent expansion of CD133 plus cells within the culture.
And CD133 is a classic marker for cancer stem cells.
Yes. The IL-6 signal physically forced the cells to transform, acquiring highly aggressive stem-like properties that we associate with rapid tumor initiation and resistance to therapy.
That is incredible. It is a phenomenal demonstration of the entire pipeline.
It really validates the whole approach.
Let's do our final recap of this journey before we wrap up. Bullet 1.
Yeah.
We took Renoir into the real world. In the mouse brain, it mapped 20 different regions purely by eavesdropping on cellular dialects, proving astrocytes talk differently depending on their zip code.
Right.
Bullet 2. In breast cancer, it mapped the tumor's actual defense architecture, the supply lines and the chemical walls blocking the immune system. And bullet 3. In liver cancer, it didn't just map the tumor. It predicted exactly how the cancer was corrupting nearby stem cells using IL-6, and then they literally proved it in a lab by watching the cells turn into aggressive cancer stem cells.
It perfectly encapsulates why the field is moving so aggressively towards spatial multi-omics.
Yeah.
I mean, tools like Renoir give us the ability to look at a tissue not just as a bag of cells, but as an active, spatially constrained society. It proves that the physical location of a cell and the exact concentration of the signals in its immediate vicinity govern its behavior just as much as its underlying genome.
It's not just who you are, it's who your neighbors are. When you look at the breadth of what we've unpacked from your sources today, from mapping the discrete layers of the cortex based purely on astrocyte chatter, to tracing the supply lines that breast cancer uses, to literally predicting liver cancer spread, it completely reframes how you look at biology. We are no longer limited to identifying that a pathway is broken. We can pinpoint the exact spatial coordinates where the faulty commands are being issued and executed.
And the clinical horizon for this is vast. As spatial resolution continues to push toward true subcellular levels, frameworks that integrate these receptor constraints in information theory, they will be the standard for discovering novel therapeutic targets that are entirely invisible to traditional sequencing.
Which leaves you with a really profound realization about the sheer scale of the microscopic logistics happening inside you right now. I mean, if a tumor can survive by strategically waking up archived fetal communication pathways to corrupt its neighbors, what other dormant cellular networks exist within our tissues? Are there completely unknown localized neighborhoods of cells in our bodies right now, just waiting for the right or wrong molecular text message to wake up and fundamentally alter their identity?
It's a slightly terrifying but mostly amazing thought.
It changes how you view every biological process, knowing that it's all dictated by this hyper-localized, highly regulated spatial dialogue. Thank you for bringing these sources and joining us on this deep dive. And to you listening, something to chew on the next time you think about the trillions of cells talking inside you right now. Until next time, keep questioning the map.